in humans, there's only three basic kinds of muscle. So muscle in humans is classified this way. First, it's on whether the light microscopic appearance of these cells or fibers is either striated or non-striated. Okay. And then you divide the striated variety into two subcategories. One called skeletal, which is mainly found in the limbs and the core, or the trunk of the body. And the other is cardiac muscle, which is pretty much found only in the heart, rarely in the beginnings of the great vessels, but pretty much confined to the heart. So here's kind of an easy table if you want to beginning making a study sheet of drawing or practice drawing it. I think it'll help you remember the characteristic features of these three muscle types. So in the top panel, we've got a kind of striated muscle called skeletal. In skeletal muscle, what you have are these long cylinders, long cylindrical fiber type structures that are multinucleated. So it makes sense to not really call these individual cells, but to call them fibers for that reason, because they're multinucleated. If you look at, and they're shown in both a longitudinal and a cross-sectional image here. If you look at the longitudinal image, you can tell that the nuclei are located just under what would be the plasma membrane of each of these muscle fibers. So that's very characteristic. Multinucleated, and the nuclei are found underneath the plasma membrane at the edge or the periphery of each fiber. If you would look at them in a cross-section, that panel to the right, you would see that in many sections, in many of the cross-sections, you could find nuclei, and the location is in the characteristic place that's at the edge of the muscle fiber. Because of the vagaries of the section, you don't know exactly where you are when you ever make a cross-section through a group of muscle fibers. You're not going to catch the nucleus in every fiber. That's just the way it goes. So yeah, don't worry about those. Worry about the ones where you can actually see the position of the nucleus. In skeletal muscle, you'll notice that in cross-section, they're pretty rounded. Most of them are, for the most part, rounded, or I guess you could say sort of trapezoidal in shape, elliptical maybe. So these skeletal muscle fibers, if you want to think of an analogy, are arranged a little bit like the pipes on a flatbed truck. They don't all sit parallel to one another. They're sort of arranged in a stacked random configuration, so that in any longitudinal section, you're going to catch some right in the thickness of the muscle fiber, but in others you might catch only the top or the bottom. In other words, a tangential longitudinal section, it wouldn't be as thick. And that's probably what accounts for this slight variation in its cross-section appearance. Okay, so much for skeletal muscle. The middle panel shows cardiac muscle. Again, this is a kind of striated muscle. The big difference between it and cardiac is that these are individual cells. They're not a multinucleated fiber. They're an individual cell. The position of the nucleus in each cell is pretty much in the center of the cell. And each cell has a definite beginning and end, has this kind of a Z-shaped or jagged line. We'll show you here, or here, or here. Those are called intercalated discs. So each cardiac muscle cell begins and ends. There's no way to say which end is the beginning, which is the ending. It doesn't really matter. It stops as an intercalated disc. So big differences right away. Individual cells, not multinucleated fibers. The position of the nucleus is different, in the center of the cell, not at the periphery or the edge. And cardiac muscle fibers or cells branch, they split. That's why, again, if you see them in cross-section, you'll have probably a greater variety of shapes. Some will be nice and round. Some will have the nucleus right in the center of the cross-section, but there'll be plenty of cross-sections where there is no nucleus, again, just to the randomness of the section. And in some cases, you'll see these little tails or tags or horn-like pieces coming off. Those are probably pretty close to a branch point. Okay, so that's the big differences between skeletal and cardiac muscle. The bottom panel shows you a longitudinal and a cross-sectional appearance of smooth muscle. Again, these are individual cells. They have a nucleus in the center. They are not striated, Yes, they do contain actin and myosin, myofilaments in them, but they're not in registration, so they don't have a striate appearance. And whether you see them in a, whether you see them in a longitudinal section or a cross section, again, the position of the nucleus and the fact that they're non-striated will help you recognize them. Mainly smooth muscle is confined to the visceral systems of the body, like respiratory, reproductive, urinary, gastrointestinal, where you need a long, rather slow, continuous contraction of sheets of these cells. And for that reason, if you look on the right-hand edge here, they are under involuntary control. They're controlled by the autonomic nervous system, their contraction. The other two muscle fiber types, skeletal muscle is under your control, hopefully. It's adapted for rather strong, quick contractions, but discontinuous, not prolonged over lengths of time. And then cardiac muscle, again, strong and quick, rather would appear to be continuous contractions. That's probably not exactly true, but again, not under your control. It's controlled by the autonomic nervous system, so it's under involuntary control. Now, before somebody raises the issue and says, wait a second, what about a muscle like the diaphragm, which is skeletal muscle? And you do have a certain level of control over the contraction of your diaphragm, but there are various centers in the brainstem that control the initiation of contraction of the diaphragm. So yes, even if you hold your breath, you could probably hold your breath until you pass out, but after you hit the ground, those respiratory centers would take over and you'd start breathing again. So that's kind of an exception. So if you're trying to make a study sheet, this is a good place to start for the muscle tissue lecture. So in general, skeletal muscle, a text of what I said, just for those of you who keep emailing me and saying, yeah, I like those text summaries, okay. Now, parallel groups of muscle fibers, they're surrounded and sort of held together, or separated sometimes by connective tissue. Almost all skeletal muscle has an origin and an insertion as a tendon. So most of these are muscles that come from or insert on bone. The place where they come from is called the origin. The place where they eventually insert or terminate is called the insertion. They need an abundant vascular supply because they're very metabolically active. And they have to be richly innervated by both motor and sensory neurons if they're going to do what they're supposed to do. Some actual images of skeletal muscle. The panel on the left is again a longitudinal section. So you see various multinucleated cylinders of skeletal muscle here. In some, you're getting probably the full thickness of diameter. In others, just getting a slice of the top or bottom. So that's why there's a variation in their diameters. Most of the nuclei you see at the edge are probably nuclei that belong to the skeletal muscle fiber, but there is another group of cells in their nuclei that are rather inactive for the most part, unless they're called upon, that will look very similar. So the only question I could ever ask you at the light microscopic level would be to, what do you think this nucleus belongs to? You would have to say the skeletal muscle fiber. We'll talk about another group of cells that are in that position, but you really can't identify clearly at the light microscopic level. There's a picture of a capillary here with probably some stacked red blood cells in it. Not sure if this is a capillary, probably is here. The capillaries run parallel to the longitudinal axis of these muscle fibers. So that means in a cross section, which is what you see here, they've obviously got these cross sections of skeletal muscle fibers. Nuclei, multinucleated nuclei lie at the edge or the periphery of each fiber. And these things that are marked by the asterisks, those are cross sections of capillaries. Our capillaries also show a cross section, which makes sense. There is a little bit of connective tissue in between or among these muscle fibers. These are muscle fibers. So yeah, there might be an occasional fibroblast in here making the connective tissue fibers that we've talked about connective tissue, but by and large, those are rather insignificant. They're there. We'll talk about them as a layer that surrounds each muscle fiber, but for right now, I'm not going to emphasize their importance. They are important in one way when we talk about muscle fiber injury. Okay. You have to understand that what we see in our tissue sections represent adult skeletal muscle fibers. We don't have any images of embryonic or fetal muscle. And the reason is that muscle fibers start out as individual cells, as myoblasts or myogenic precursor cells that eventually receive a certain set of signals to first commit them to skeletal muscle, or actually striated muscle differentiation, and then later down the line, skeletal muscle or cardiac muscle differentiation. So first what you get are individual cells that become myoblasts. They receive certain signals. The MRF stands for myogenic regulatory factors. There are a bunch of these. MyoD is one of them. PAX3 is a paired box transcription set of factors that sort of commit these cells to a certain line of differentiation. And then when they fuse together, you get this thing called a myotube. So a myotube is sort of the real beginning of a multinucleated skeletal muscle or striated muscle fiber. Myogenin is another transcription factor and marker of myotube differentiation along a striated muscle fiber course. So it's more specific than some of these general regulatory or transcription factors that appear earlier in development. So that's how you get a multinucleated fiber. These start out as individual cells, become myoblasts. The myoblasts fuse to form a myotube, and then that myotube elongates and differentiates into a mature skeletal muscle fiber. You guys talked about this in embryology, I assume? Yes? No? No? Okay. You guys say yes, okay. You guys have had myotomal differentiation, paraxial muscle myotomes and stuff like that in embryo so far, yes?
Yesterday.
Yesterday, okay. Maybe Dr. Martin will talk about it yet, okay. Okay, now I'm gonna switch gears just for a second. I'll go back to that sort of theme in a minute, but I need to define something for you for the rest of this to make sense. And that's the definition of a motor unit. A motor unit is defined this way. It's the motor neuron, a multipolar neuron, its axon, and all the skeletal muscle fibers that axon innervates. That's a motor unit. A motor unit is not the thing under the hood of the car that makes it go. A motor unit is a motor neuron, its axon, and all the skeletal muscle fibers it innervates. So if you look at it in sort of a teleological frame or sense, a single motor neuron can contact or innervate several hundred of muscle fibers. It's been determined and shown pretty clearly that muscles that are capable of really delicate, very precise movements, like extrinsic muscles of the eye, very delicate muscles of the hand, especially extrinsic muscles of the globe of the eye, have one motor neuron, its axon innervates only a few muscle fibers. Muscles that have really gross or less precise movements, like especially postural back muscles, deep back muscles, superficial back muscles, have a single neuron that innervates maybe hundreds or thousands of muscle fibers. So again, if you want really precise movements, you need to have only a few muscle fibers innervated by one motor neuron. When that motor unit meets or innervates a group of muscle fibers, those terminations are called motor end-plates or neuromuscular junctions. So here's an example of kind of maybe 5 or 6 of these, what they look like. So what you get is, this is probably a number of axons in this thing called a nerve here. And they kind of split apart to innervate individual muscle fibers at motor end-plates. Okay, and the neurotransmitter released there is acetylcholine. It's important that you remember eventually, if I ask you, not only does the motor neuron cause the muscle to contract, it certainly does that, but it also exerts what's called a trophic influence on all the muscle fibers in a motor unit. That means that if that motor neuron or its axon is lost, or the axon's cut, it doesn't grow back, that muscle fiber will probably undergo atrophy, called denervation atrophy. It's been separated from its innervation, and it can't exist alone. It will undergo atrophy. So the term that's used to describe that is denervation atrophy. So if you've seen someone who's had a spinal cord lesion, a gunshot wound, a knife wound, or a stroke, cerebrovascular accident. If neurons are lost because of the accident or trauma, all the muscle fibers that were innervated by them are going to eventually atrophy. So you get these people with flexure contractures and stuff like that, because now what were flexors matched by sensors in the forearm or hand, not only one set of muscles are acting, either in extension or in flexion. So you get these contractures. Okay, here's an example of what I'm trying to say. So in the image to the left, we've got a multipolar neuron here. Let's say in the gray matter of the anterior horn spinal cord. It's got a single axon, it's myelinated. It comes out, you know the cells that myelate it in the CNS, oligodendrocytes, and do the myelination in the PNS, Schwann cells. Contacts a muscle fiber at its neuromuscular junction here, a motor end plate, and innervates the muscle. It also causes it to exist. It's a trophic influence. If that axon is injured, or if we just destroy the nerve cell, and we don't make the connection, we get denervation of that muscle fiber. That we don't get a connection anymore. So if you hear of, for you guys who working at scribes in ERs, like Brian, you know, if he sees somebody come in with a serious traumatic injury, a bad car accident, a gunshot wound to a limb, even if the neurosurgeons would reconnect as many as possible nerves in a nerve bundle, I don't know, like musculocutaneous nerve, median nerve in the upper limb, because you've got both sensory and motor neurons that are involved in that nerve, you're hardly ever going to get 100% full return of function, because the sensory connections have to match the sensory terminals, the motor connections have to match the motor terminals, the motor end plates. In a nerve, there's no 12 o'clock, 6 o'clock, 3 o'clock, 9 o'clock position on the nerve, so the neurosurgeon will try and match it up as best he or she can, but you're never going to get 100% full functional recovery, unfortunately. Yeah. So just something to think about. All right, let's talk a bit about the connective tissues involved in skeletal muscle. There's 3 different layers or sort of flavors of connective tissue when we talk about skeletal muscle. First, the one that I've kind of showed you or hinted at already is called the endomysium. That's how you pronounce that term. It's the really delicate, very thin, loose connective tissue that surrounds each individual skeletal muscle fiber. That's called the endomysium. On that cross section, I showed you, a couple of fibroblast nuclei, a little bit of connective tissue around each muscle fiber. We've got muscle fibers here. So this very delicate connective tissue in here, it's really hard to see with an H&E because there's just not much there. That's the endomysium. Okay. When you get groups of muscle fibers sort of banded together, that's called a muscle fascicle. And there's another different layer of connective tissue around muscle fascicles, that's called the perimysium. So endomysium around individual muscle fibers, perimysium around groups of muscle fibers called fascicles. And then of course there's the epimysium or connective tissue that surrounds the muscle as a whole thing. That's the epimysium. It's going to be the thickest of all these connective tissue layers associated with skeletal muscle. Here's a picture of that. It's sort of a cutaway diagram of a whole muscle. We'll just pick one in the arm like the biceps. So the connective tissue that's very thick surrounds the muscle all the way around it on the way outside, externally. That's the epimysium. These sort of segregated groups of muscle fibers here, those are muscle fascicles, and the perimysium is surrounding each of those. That's the perimysium surrounding a group of muscle fascicles. But individual muscle fibers, as are pictured here, has a very delicate little bit of connective tissue, sort of pictured in yellow, pale yellow color. That's the endomysium. Okay, so the endomysium is this really delicate, thin layer of connective tissue that surrounds each individual muscle fiber. I mentioned earlier that almost all skeletal muscles, and I'm trying to think of a couple that don't, and I can't think right now. what's for lunch maybe, begin and end as tendons. And you might say, well, wait a second, how's that work? Well, a tendon is essentially a connective tissue structure that anchors skeletal muscle to a bone. And that's how almost all skeletal muscles work. There's a couple, I can think of a couple maybe muscles of the orbit that don't work that way, but those are the exceptions. So what we have here is the termination of a group of muscle fibers in a muscle. Some are bound together in a group of, like a fascicle, so the connective tissue perimysium surrounds a muscle fascicle bundle. Each individual muscle fiber is surrounded by a delicate bit of connective tissue, that's the endomysium, and eventually they all sort of stop. This is a bit artifactual. They actually sort of terminate like the tips of your fingers, and they interdigitate with the beginnings of the tendon, sort of in a dovetail fashion like that. Okay. And then the tendon attaches to the periosteum that surrounds the bone. Okay. When you hear about, and this is called a muscle tendon or myotendinous junction, when you hear about this structure, it is very stable. There is a belief that in adults, you cannot separate, in a normal, healthy adult, you cannot separate the muscle from its tendon. So when you hear of somebody having a muscle pull or a muscle separation, the muscle isn't actually separating from the tendon. Either the muscle fibers themselves are splitting apart, or you're pulling the tendon off the bone. Yeah, and that takes some doing. It usually occurs in very large bellied muscles that narrow to a very specific point on a bone, like the muscles in your calf. You ever look at a sprinter, when a sprinter pulls a muscle, especially those guys in the 100-yard dashes, they have these huge group of muscles in the back of their calf that narrow to a point on their calcaneus bone. So when they tear a muscle, they're tearing that Achilles tendon off the bone, which in addition to being really painful, is quite disfiguring as well, because then you have to surgically reattach it. Yeah. The only reason I know anything about this is because this was my research for my PhD. Yeah, I put animals on a diet that solubilized the collagen in the muscle tendon junction and tried to see if we could get these animals to separate their myotendinous junctions. That was part of it, and also to figure out which cells are making the collagen and what happens to these muscle tendon junctions when they're disturbed, like when the collagen is more solubilized. You cannot, I could never get, in the 80 animals I did, both sides, both limbs, I could never get a muscle tendon junction to fail. So they are very, very strong. In kids, sometimes you can get a muscle and its tendon to separate a bit, but not much, it's pretty hard to do that. So it's a very stable structure. Here's what it looks like on an electron microscopic image. These aren't images from my PhD thesis, but they're pretty similar. What you have are these terminations of the skeletal muscle fiber coming down here on the left, kind of interdigitating with the connective tissue components and cells of the tendon a little below that. Again, these terminate like the fingers interdigitating on your hand. Yeah, so a lot of different proteins are involved in this, mainly type 1 collagen, but proteins that span the membrane of the muscle fiber to make those connections with the collagenous parts of the tendon. Yeah, if you want to read a really boring PhD thesis, I can let you read mine. It'll put you to sleep in about 5 minutes. Yeah, it's about that thick. I believe that's called packing of literature. So let's summarize now what we know, what I told you about skeletal muscle fibers. So extremely long, especially on a microscopic scale. You know, the longest muscle in the body is the muscle that comes sort of down your medial aspect of your thigh here, called the sartorius. Some of those muscle fibers are foot and a half in length. So on a microscopic scale, those are really very, very long. Muscle fibers are multinucleated, so they should really be called a syncytium, if you like that term. In other words, they're not a single cell. The nuclei are located at the edge of the muscle fiber in a peripheral position. And because people who study muscle think they're pretty special, they feel it's okay to give the muscle plasma membrane a separate name. It's called the sarcolemma. And they feel like it's okay to give the cytoplasm of the muscle fiber a different name too. They call it the sarcoplasm. So sarcolemma is just nothing more than the plasma membrane of the muscle fiber, sarcoplasm, cytoplasm. Basically, contractile filaments of actin and myosin. There is a basal lamina, or basal membrane, whatever term you prefer, that's outside the plasma membrane of a muscle fiber. And within that basal lamina material, there is another group of cells. These are the cells I said you could not identify at the light microscopic level. They're called satellite cells. So these satellite cells, basically all you see is their nuclei, because for the most part, they're lineage-specific stem cells. They live most of their life within the basal lamina of a muscle fiber, skeletal muscle fiber. How could you tell them apart? How could you distinguish them? Good question. Here's the way you do that. You can only do it on a electron micrograph, I think, for our purposes. So on this image, there's two skeletal muscle fibers. On the run to the right, you see the nucleus of the skeletal muscle fiber in this eccentric or peripheral position. It's labeled number 1. So just outside of it would be the plasma membrane, the sarcolemma of the muscle fiber, labeled number 4. The stuff, labeled 3, would be sarcoplasm, mitochondria, myofilaments of actin and myosin within the muscle fiber. So number 1 represents the nucleus of that particular fiber. There's another muscle fiber off on the left side here. We don't see its nucleus, but we do see a rather quiescent looking cell here, labeled number 7. So what do we see? A nucleus, a lot of heterochromatin, again, makes you think, probably not much going on with it, hardly any cytoplasm, and it is contained within the basal lamina material of the skeletal muscle fiber, labeled number 10. The basal lamina material is probably labeled number 9. So these are satellite cells, mainly demonstrable by their nuclei, because they don't have much in the way of cytoplasmic features, and they kind of sit there. They act as a stem cell. So when there's an injury to a skeletal muscle fiber, these satellite cells receive a group of signals. Some of the signals are very similar to what we discussed when we talked about myoblasts differentiating and forming skeletal muscle fibers. And they basically now control the segment of the muscle fiber that's been injured or damaged. This system works especially well if the injury to the muscle fiber has not completely destroyed large sections of the muscle fiber, and if the basal membrane material remains relatively intact. These satellite cells undergo a process of differentiation. At one point, I'm talking about 25 years ago, I think, it was believed that the satellite nucleus just sort of dove into and became a part of the localized part of the skeletal muscle fiber. Now it's believed that the satellite cell nucleus undergoes a bit of a proliferative activity. It undergoes what's called asymmetric division. Anybody know what that is? Asymmetric division? Okay, I'll explain in a second. I can remember what it is. It sort of takes control, acts as a nucleus of the skeletal muscle fiber. All right. So here's kind of an image of how that happens. So in the left-hand side of this panel here, this image, got a number of multinucleated skeletal muscle fibers, and their nuclei are in blue, pale blue. The nuclei in green is a satellite cell nucleus. So that's this guy here. So with injury or activation, we get what's called asymmetric division. Asymmetric division means that normally in a symmetric division, you get 2 daughter cells, and they both are equivalent in their eventual arc, career arc, locational arc. In an asymmetric division, one of these satellite cells becomes another satellite cell and assumes that quiescent appearance. The other one appears to undergo sort of myoblast differentiation, and that's what fuses with the damaged segment of the muscle fibers that we talked about earlier. So it's believed that's how this occurs nowadays. This satellite cell doesn't simply merge with the localized damaged region of a fiber. It undergoes what's called asymmetric division. It produces first, or at least, another satellite cell and a myoblast, which fuses with the muscle fiber. If you're adding bulk to muscle, it's believed that's the same thing occurs. You add bulk to muscle through exercise and activation, at least in adults, of satellite cells with asymmetric division, infusion of myoblast cells to existing muscle fibers to increase their diameters. Okay. Let's talk now about red and white fibers. And this, I'm sure this will come up in your physiology class, at least I think it will. Ever since the beginning of the point where people made microscopes good enough to look at tissue sections, and you developed apparatuses, microtomes, that could cut pieces of tissue thin enough to look at under a microscope, that transmitted light so you can actually see something. Ever since that time, it's been known that if you look at a cross-section of any group of skeletal muscle fibers, cross-section, they're not all the same. The big differences are, there are sometimes pretty big differences in their diameters. So if you look at, if we ask Brian to give us a section of a deltoid muscle, yeah, we're not going to ask him to do that because you probably refuse. Hey, keep your scalpels out. And we froze it, because you got to cut it. It's got to be hard enough to cut with a microtome. And we looked at it in cross section. You could see differences. Some would be very narrow in diameter, some would be much larger in diameter. So those differences are called now red and white fibers. And they're first noticed on the basis of cross-sectional diameter. This sounds like a research project for a graduate student, doesn't it? Yeah. I want you to look at these muscle fibers, these 10,000 sections of muscle fibers. And
you got to section them first.
Yeah, tell me what you think. Okay. So one of the ways to do that, a more modern way to do that, is to make a frozen section. So you take a piece of tissue, you freeze it, you cut it on an instrument called a cryostat, which keeps it cold, so it's hard enough to be cut with a very sharp knife. And then you layer on it a variety of different substrates that will be acted upon by the enzymes in the frozen tissue. Okay, so this is called histochemistry. So this is a histochemical preparation of a random muscle, we'll just call it the deltoid or biceps muscle, with some substrate that's going to be acted upon by enzymes in each of those muscle fibers, and hopefully eventually produce a colored product so we could distinguish them easy. All right, as you look at this, you can say, well, some of these are pretty narrow in position. Like these guys that are darker stained with whatever this process is, and some of them are much lighter in position, lighter in color, and larger in diameter. So we call these that are larger in diameter type 2 fibers, and these that are more narrow in position, type 1 fibers are red fibers. Okay, so here's the detail, a little bit of detail on what each of those represent, which of these fiber types represent. Red fibers are called slow twitch or type 1. They're the ones that were smaller in diameter. At the tissue level, they contain relatively large numbers of mitochondria and large amounts of myoglobin. They display strong enzyme reaction products like NADH or succinic acid dehydrogenase activity, again, whatever substrate you layer on there first, to produce a colored product. They're called slow twitch. It means that they have a better resistance to fatigue, so they're adapted for rather long, slow contraction. They generate relatively less tension, but they don't fatigue as quickly. So I guess if you were a marathon